Welcome to the topic 2 on our lecture series on the mechanism for antimicrobial resistance. So during this topic 2, we will mainly discuss about mutations. So in our topic 1, we discussed about selective pressure, and in the topic 2, we will discuss about mutation. Just to recap on selective pressure, you will remember that you have a microbial population, and within that population, there are some bacterial cells that carry resistant gene. And when you introduce an antibiotic, that antibiotic will kill the majority of the bacterial cell, but those that carry resistant gene will remain and will then have enough space and a nutrient to continue to replicate, and they will then overpower the rest of the population. So the next time that you want to use the same antibiotic, you will not be able to succeed because you are only dealing with a resistant microbial population. Now, during mutation development, what happens is that we know that most microbes reproduce by dividing every few hours, allowing them to evolve rapidly and adapt quickly to the new environmental condition. And during that replication process, mutations can take place. So here we are in a situation where you have a microbial population. None of these carries an existing resistant gene. But now during the replication process, mutation can happen. And that mutation will then help an individual microbes to survive exposure to an antimicrobial agent. So this is what you can see here. We have our bacteria that is a non-resistant bacteria, and these start replicating. It starts replicating, and during the replication process, you will see this bacteria here experience a mutation in its DNA because of the replication process step. And you can see another one here that also develop resistance. It also develop resistance, And then what happened is that the antibiotic will then kill all the susceptible microbial population, and only the resistant one will then remain. Those that are that's just developed mutation will then remain, and they will keep replicating. So we then have only drug-resistant bacteria that keep multiplying. Okay, so this is the result, development of resistance through mutation. So the difference with selective pressure, I repeat again, in the selective pressure, the microbial population has some bacterial cell that already carry an existing resistant gene. But during the mutation process, you have a microbial population, and during the replication process, there are then some bacterial cells that start mutating. So their mutations start taking place in their DNA. Then they will then develop resistance, and they will resist the effect of the antibiotic that is being used. So that's the difference between selective pressure and mutation. So microbes also may get genes from each other, So there is also an exchange of the genes between microorganisms, including genes that make the microbe to become drug resistant. So those bacteria, they usually multiply by the billions, and they have then drug-resistant DNA that transfer a copy of these genes to other bacteria. So non-resistant bacteria will then receive the new gene, the new DNA that has been mutated, and become resistant to the used drugs. So in the presence of antibiotics, only drug-resistant bacteria will then survive. So the drug-resistant bacteria will survive, will multiply, and will continue to thrive. So this is what we illustrate here. So you can see that you have your original bacterial population, then you add an antibiotic like penicillin, then this penicillin will then kill all the other susceptible pathogens, those that are susceptible to penicillin. But the strand or the bacterial cell that mutated from here is the one that will remain, and the mutant one will then replicate, and later on we have a microbial population that is only made with resistant bacteria. So that effect of mutation can then be manifested in several ways. So one of the ways we can see the manifestation of the effect of mutation is that we can have what we call the altered permeability. So here you can see the cell membrane of a bacteria. Then because of the mutation, the permeability of the bacterial cell will be altered. And an example of altered permeability is what we call altered efflux. So there is here porins, and there is an effect of efflux that can be altered. We have what we call efflux pump that can be manifested, and it will start pumping out all drugs that are coming in. It's pumping it outside the cell. And this is one of the mechanisms of resistance against the tetracycline antibiotics. So when you use any of the tetracycline antibiotics, and one of the resistant mechanisms against tetracycline is the development of efflux pump. So there is altered permeability, development of efflux pump that will take all the tetracycline drugs outside the bacterial cell. Another effect of mutation can be the development of enzymes like beta-lactamase that inactivate the drugs. Mainly, your beta-lactam drugs are affected by the enzyme beta-lactamase. So those beta-lactamase are a result of mutation that take place into the bacterial DNA. Another enzyme that can be produced that can lead to resistance, for instance, to chloramphenicol drugs, it's called, it is the enzyme chloramphenicol acetyltransferase. So the development of chloramphenicol, the activity of chloramphenicol acetyltransferase can be, can develop as a result of mutations, and it can destroy the effect of antibiotics like your chloramphenicol. So the other effect of mutation is what we call altered target site. We know that antibiotics, for them to act, they need to bind themselves somewhere. And a good example is always of penicillin or beta-lactam drug that binds to what we call PBP, penicillin binding proteins. Now, when you have a mutation, that mutation will then alter that target, that PBP will change. There will be changes in that PBP. And as a result, the beta-lactam that is given will not be able to attach appropriately with that PBP. I can give an example of Staphylococcus aureus. Staphylococcus aureus, for example, how it becomes resistant to all the other penicillin drugs. You know, there is a gene that we call MEC2A gene. So when you have a mutation in the MEC2A gene, there is alteration of the PBP in the Staphylococcus aureus, and there is what we call a PBP2A that develop, and your penicillin will not be able to attach to the new PBP. So the newly changed PBP, altered PBP. So as a result, we only use cloxacillin, for example, that is able to attach to the new PBP that is formed. So this is an example of altered target site. It's the same thing like antibiotics like fluoroquinolone, like your rifampicin, for example, that target RNA polymerase, or we have your aminoglycoside that target ribosomes, that target ribosomes. So if there is an alteration of the target site, that is a TTS subunit of the ribosome, then they will be, we will not have a good interaction between the antibiotic and the target of that microorganism, of that antibiotic, that is TTS ribosome, like your aminoglycoside. They target the TTS ribosome. So they will not be able to do so, and they will be resistance development. So this is what we try to summarize here. We can show that there might be mutations in bacterial topoisomerase 4 and DNA gyrase that prevent fluoroquinolone from binding, allowing replication on the bacterial genome to continue. We know that your fluoroquinolones, what they do is that they bind to the topoisomerase 4 and DNA gyrase. Those are the target enzymes that they attach to, and in order to destroy the effect or to prevent the effect of fluoroquinolone. Okay, so mutations in bacterial topoisomerase 4 and DNA gyrase will then prevent fluoroquinolones from binding, and they will be resistance development. The same thing is that bacterial modifications to an efflux pump can allow the bacteria to pump fluoroquinolones out of the cell. So here you can see there is an efflux pump that is established as a result of mutations. Then consequently, we have fluoroquinolone drugs that comes inside the fluoroquinolones to try to bind to inhibit the effect of the nucleic acid, but they are subsequently pumped out of the bacterial cell. And here we can see bacterial DNA replication enzymes like topoisomerase 4 and DNA gyrase that we spoke about that are associated with resistance to fluoroquinolones, that are associated with resistance to fluoroquinolones. Okay, so we can summarize different types of mutations and their effect here. Here we can see that there is here we can see that there is a decrease porine expression in A. We have a decreased porine expression here. We have decreased porine expression, and in B, here we can see that there is a methylation of the TTS ribosomal RNA, so with a decreased antibiotic binding. An example I gave of aminoglycoside that bind to the TTS ribosomal TTS RNA subunit. So you will see that if there is a methylation of this TTS ribosomal RNA, so what will happen is that there is a decrease in antibiotic binding, especially your aminoglycoside that act here. And in C, we can see that mutation lead to a production of an enzyme like your beta-lactamase, and that will lead to an enzymatic inactivation of antibiotics, mainly our beta-lactams. And in D, here we can see that we have aminoglycoside modifying enzymes that will lead to the decreased affinity for ribosomal subunit. And this mechanism is also valid for aminoglycoside. And here E, we have the topoisomerase 4 and the DNA gyrase mutation that lead to the decreased antibiotic binding especially with the fluoroquinolones, we refer because they act on they are nucleic acid inhibitors. And in F here, you can see that there is an increased efflux pump expression. So that will pump away the antibiotics that went through inside the cell, though they are pumped out. And here, the mechanism of resistance here can be applied to beta-lactam drug, fluoroquinolones, aminoglycoside, trimethoprim, tetracycline, and chloramphenicol can develop resistance by developing an efflux pump. And G here, it's about modification to the lipopolysaccharide. So there is a decreased antibiotic binding. So that specificity to bind to the lipopolysaccharide, like for antibiotic like colistin, will be developed. So colistin developed resistance by actually decreasing antibiotic binding specificity with the lipopolysaccharide. So those are different mutations that lead to different things, and we have different antibiotics that can be affected through that particular mechanism. So let's try to look at our test one. The question is, how do bacteria acquire mutations that make them resistant to antibiotics? How do bacteria acquire mutations that make them to become resistant to antibiotics? So the answer one is it novel mutations in the DNA of a bacterial cell that lead to mechanisms that counteract effect of drug, or acquisition of new genes via sharing of DNA from other, through sharing of DNA from other bacteria, Is it A and B or none of the above? Pause your video for 30 seconds and think about the answer. So you will see that it can be either a novel mutation in the DNA can lead to mutation that make bacteria resistant to an antibiotic. So this answer is correct, but it can also be acquisition of new genes via sharing of DNA between bacteria. So this also is correct. So the correct answer is then C. It's both A and B is the correct answer. In test 2, the question is, which of the following are the effects of mutations in bacteria that allow bacteria to develop resistance to antibiotics? So which of the following are the effects of mutations in the bacteria that allow the bacteria to develop resistance to antibiotic? Pump the drug into the bacterial cell. Is that the effect of mutation? Or alter the target site where the drug usually binds to the bacteria so it can't bind anymore? Is it the effect of mutation? Produce enzymes that inactivate the antibiotics. Then D, A and B only, or E, B and C only. So pause your video for 30 seconds and think about the correct answer. Okay, so if we look at the response A, we say pump the drug into the bacterial cell. This is not true, because when we have the efflux pump that is developed as a result of a mutation, it actually pumps the drug outside the cell, not inside the cell. So this answer is wrong. Alteration of the target site is true, we say that when there is an alteration of the target site where the drug usually binds to the bacteria, then the drug cannot bind anymore because of alteration of the target site. So B is true. And C, they say production of enzymes like your beta-lactamase that inactivate the antibiotic. This is also a result of mutation. So B and C are true. So B and C are true, so the answer is E. The correct answer is E. Okay, so follow us in our topic 3 as we are now going to discuss the horizontal gene transfer. So don't miss this topic 3 because it's very, very critical and we are going to discuss the horizontal gene transfer. Thank you very much.